You got that?
No, I'm just thinking of how am I gonna say this? Because when we were having the conversation, there's like a lot of ways where you've answered other, kind of a little bit of other questions as well. So like, what is the reason you are researching these things in like in an in-depth way, and how they are being understood? Like as in, how you were talking about leukemia, and like significantly your acute myeloid, and
Okay.
yeah, how
Yeah,
you're
because see, if you look at the leukemia, what is meant by leukemia? It is an aberrations in your blood cells.
Mhm.
Because of that, the cells keep dividing.
Yeah.
There's no control.
Mm-hmm.
So your bone marrow get jammed, get out, and go to other organs.
Like
Keep dividing and
yeah.
overcome all these organs. I mean, wherever it goes.
Mm-hmm.
There are, if you look at the blood cells, there are different kind of blood cells.
White.
B lymphocytes, T lymphocytes, based on their function. One produces antibody, another produces immunity in a cell-mediated way. Otherwise, you will have myeloid origin cells. They do their function. So all these cell types, if you look at it, if it happens to be lymphocytes, we have a very good cure rate now.
Mm-hmm.
Very effective medicines are available for that. T cell, little bit difficult, some subtypes. Myeloid, you have 50% problem. There is no cure right for them. So there are large number of cases. Still, we cannot find an effective cure. That is what our lab focus is. Specifically looking at those difficult to cure patient model and why it is difficult to cure, what are the major component, and if you can identify any of those things, you could prevent that molecule's expression and prevent giving any harmful effect because of that molecules.
And how it works out in the end. What is your main purpose in collecting data? And like, is it used by other scientists? Like, how you were talking about?
So an excellent example is, see, what we generally do is you collect the sample. Say, for example, like kid is coming with leukemia to the clinic. We ask the patients, the family to consent for taking some sample from them. We collect that samples, and these samples will be very limited. So with that, you cannot do much work. So we generate models in mice. We collect that sample and inject into mice, and they develop leukemia. So like that, we have large collection of these models. Even though it is leukemia itself is very heterogeneous. If you take 10 patients, you cannot think of giving the same medicine to 10 people.
Yeah.
So that is a major problem. That is why the cancer can never be cured as of now, because of this heterogeneity. We have to add. So this is a multi-disease, honestly speaking. Though we call it as acute myeloid, leukemia for A or B or C. You will have one type, you will have another type, you will have another. Everything is myeloid only.
Yeah.
But the expression of some molecule decides whether it is going to be aggressive or not aggressive. Do you understand? And why they are aggressive in that? These are all the basic science that is going on. So once you have that model, the advantage is you can use different molecule to cure the mice. Am I right?
Yeah.
So we have used various such molecule to cure some form of leukemia, and we found out that a particular combination of molecule can completely cure the mice. Okay? So once you do that, this is called a preclinical testing. What is that?
Preclinical testing.
That means you are generating enough data in the animal work and the lab work using these cells, saying that this drug is very promising. Now what will you do? we have clinics here, so clinicians will find an interest in that. They file an IND, that is intend to develop something for a drug.
What is IND? It's for me.
That is for if you have to do some clinical trial and things like that, you have to get the FDA permission.
Oh, yeah.
So
Okay.
Food and Drug
Associated.
Administration.
Yeah.
So they will give you the approval for that. So once they are getting the approval, then they start the clinical trial. Clinical trial means they really
Bring in
use
patients.
this medicine to the patient. So already something that we developed is in clinical trial. So what was your question?
How does this process of what is the main purpose in collecting these data? Is used by scientists.
Now you understood.
Yeah.
Purpose.
Mm-hmm.
So this is an excellent example where you do something in the lab, finally end up in the clinics, and that is the ultimate purpose of any research. Am I right?
Yeah.
Any kind of research is to make the life of a human being better in any way possible.
Mm-hmm.
Am I right? He should feel good. So that can be in medicine or in agriculture or in computer science, anything.
Yeah.
Make his life better.
Mm-hmm.
Possibly the atmosphere also. Okay. You are done?
No, I have like 2 more questions,
Okay,
but they're minor.
good.
This is more of a personal question. This really doesn't have anything. Like, how
Mhm.
does this process of sharing and collecting information work between like scientists, and how does it like move? I just wanted to know about that myself.
Honestly speaking, there is no sharing data. Is for example, I am working on something here. Then my colleagues in Germany is interested because he is pursuing on related area.
Yeah.
So this data that I find here in a particular disease may be helpful for him to pursue similar
Yeah.
diseases, but not that related. And
Mhm.
see, he is seeing similar changes there also.
Yeah.
So this is how this kind of data sharing is useful. He can quicken his discovery process.
Mm-hmm.
So this data sharing and having a collaborative networking atmosphere is extremely important, but you need to be having the trust. What is that?
Trust between them.
So that is something that is very important. Trusting different people and sharing data and using such data for productive discovery.
And is it just by relationships with other people, like how you were talking about, like how we're having a good
Say for
relationship?
example, I am reading a science paper today, say for example, and immediately I found Harvard A or B identified as something, and if that I can get it, I will be able to use it for my work. I immediately telephone him and ask, can you share that for me? This is a purpose, and I give the co-authorship for him in the publication. Or they will be patenting the data, or if it is patentable, you get together and patent together and things like that.
Okay. And are there any current researches going on right now? And if so, what is the reason behind it? What's the end goal of it?
Yeah, that's what current research going on is understanding why you have different spectrum of acute myeloid leukemia. What are the different reasons for the aggressiveness of this disease? And can we identify any novel molecules that causes this aggressiveness so that you can block it?
Mm-hmm.
That is called a drug.
Yeah.
Am I right?
Yeah.
That's all what the entire people are running for. Even if I don't do, somebody else will do. Okay, don't think that I am doing great. If you don't do it, somebody else will do it. So, end of the day, we are all working hard for that. So,
And you said aggressiveness. Is there like this? I can just stop it.